All right, so this is where video 1 left off. I hadn't put the little thing in yet, so sorry for that. So we're talking about enzyme specificity. So that's the idea that enzymes are shaped so that their active sites are only going to accept one type of substrate. So each enzyme only works on one type of molecule. For example, sucrase will only break down sucrose, meaning that the enzyme, if we imagine that this green enzyme right here is sucrase and that yellow one is sucrose, they're the only two that are going to bond together. Lactase is only going to break down lactose. So each enzyme is the right shape for just one substrate. Lactase will never be able to break down sucrose. Sucrase will never be able to break down lactose. So they only work for their one type of molecule they can break down. They're specific. The body's going to require thousands of different chemical reactions to live. So we have all these nice people here, like yourselves. In order for you to continue living, sitting here, listening to this video right now, you need thousands of chemical reactions to be occurring just to keep you sitting upright and your hearing working. Which means that you're going to be thousands of different enzymes in your body. All of those chemical reactions, if we were just to wait for the reactants to bump into each other so that a chemical reaction could occur that is necessary to keep you alive, we would not live very long. We need enzymes to go in there, pick out the reactants, speed up these chemical reactions, build our products quickly. So one enzyme type for each type of chemical reaction, meaning that an enzyme, in addition to being specific to its substrate, so what it attaches to, it's also going to do a specific job. So in the top right-hand corner, we have a blue and an orange enzyme, and this one is going to attach to the orange substrate, and it's going to split that apart into 2 parts. Now, each enzyme is going to do a different job. So these ones right here, these examples are showing you enzymes that are creating products where they're breaking the substrate apart. But it's important to realize that you all have just as many enzymes in your body that are going to take 2 different substrates and put them together to make one product. So we have enzymes that combine things as well as break them apart. Enzymes are also reusable. We only need a small amount of enzymes in our body because those small enzymes can be used over and over and over again. In a chemical reaction, the product will change. The substrate will change. So that molecule that's going to bond to the active site of the enzyme, that is going to change at the end of it. The enzyme stays exactly the same shape, meaning as soon as that substrate breaks off and the reaction is over, a new one can come over and bond to that active site and it can do it the same thing over again. So here's a nice video of it happening. So you start with your blue substrate, it goes in, changes to the products. Another blue one would come over, changes to products. Notice how our red enzyme is not changing. It works every single time on the product, which is why we only need not as many of them. We don't need too many enzymes. Small amount of enzymes will do a lot of work. All right, quick quiz. So enzymes are going to work by lowering what? Good. Hopefully you get that they work by lowering activation energy. So it requires less energy to get a chemical reaction going. So it's going to be faster. It's going to go quicker. When an enzyme catalyzes a reaction, the reactant is also called a product, a substrate, an enzyme, or a cofactor. Good. Hopefully you were able to get that it's a substrate. Remember, substrates go in, they're going to join with your enzymes. This is also called, so that pointing to that little hill right there that reactants need to overcome in order to get formed into products. Is that stoppage, reaction rate, activation energy, or the primary hillock? That is your activation energy. So that graph right there is showing you how much energy needs to go into the reactants to break those bonds in your reactants so that they can be reformed in the products. That's activation energy. After catalyzing a chemical reaction, the enzyme A can go ahead and catalyze another reaction. B is broken down, its atoms are recycled, or C requires a long refractory or resting period in which to rest before catalyzing another reaction. Good. It can go right ahead and catalyze another reaction. There's no breaks for enzymes. You don't need that many of them because they're going to be able to do a reaction, do another reaction, do another reaction. They're not changed or destroyed. That site right there. This is called the A, substrate mirror, B, locksmith nook, C, active site, or D, enzymatic spot. Good, I almost slipped and gave you the answer. That's your active site. So that's the location where your substrate attaches to the enzyme. Active site. All right, so this is where we're going to take a break. Go back, review if you need to, make sure your notes are filled in, and we will finish this next time.